Hey, bio. So just a couple of things about the worksheet. I just want to let you guys know that the expectations are everything be filled in, that your analysis on the front, so your protein synthesis simulation, like all those steps, the group members, um, the only place here would be like where I did not sign, and so for cleanup, don't worry about that. And the polypeptide check, um, I went ahead and checked each of those for you guys. We got it figured out as a group. So, um, but all of this needs to be filled out correctly. And then these are your analysis questions. So number one, let's see, pretty simple. Um, Number two, got to describe these in complete sentences. Okay, so what is a gene? That's a pretty straightforward question. How is the information in a gene stored? How is this information expressed? Is the way the gene is expressed the same as how the information is stored? Answer that question by explaining the difference between the two. Check with other groups in the class what variants of the gene existed. So you could see yesterday what was the difference, and then if you want to check with the other groups, you can see where that came from. And take a look at the base pairs. Could the same messenger RNA be used again by passing it to the next station? Can this happen in a cell? You can look that one up. Is mRNA used more than once? And can a specific transfer RNA pick up different ingredients? And explain why or why not. Can a transfer RNA be used again? Could it go pick up another amino acid? And then, were all the transfer RNA molecules involved in the production of this polypeptide? Um, I guess just looking at what you saw on the back counter, that would answer that question, right? And then were all the amino acids present in the room used to make this polypeptide? No, there are lots of different combinations that it could be, right? If an essential amino acid were not present in the person's diet, could all proteins be made? If you are missing one of those essential amino acids, you would not be able to make an amino acid. So just come up with something like if you didn't have this essential amino acid, then the protein would be different. So just make sure you explain using full sentences. And then in your conclusion, I am looking for 2 to 3 paragraphs that summarize the activity. And in that, you're supposed to explain how this is an analogy for protein synthesis. So be sure that you talk about the structure of DNA and how it actually determines the structure of the protein, and then how the protein supports essential life functions and the specialized jobs of cells. Just hang on a second. Let me yell at Alexa. Alexa, stop. Make sure to use the following terms. So you need in your explanation DNA, RNA, messenger RNA, rRNA, tRNA, codon, anticodon, code, nucleotide, ribosome, amino acid, polypeptide, and protein. Just highlight the words in the writing so that just the first time you use it, it needs to be highlighted so that I can recognize that that's there. If you need more writing room, you'll have to go onto the page that this should be glued down in your book as a flip. That would be 99, right?